Tetrameric structure of ionotropic glutamate receptors, AMPA receptor GluA2 homo-tetramer. Switch of dimer partners from the amino-terminal domain to the ligand-binding domain. Red, yellow and blue-green pairs at the amino-terminal domain at the top, red, blue and yellow-green pairs at the ligand-binding domain at the center. Amino-terminal domain dimer is homologous to mGluR ligand-binding domain but lacks the glutamate binding site. Small dimer-dimer interface between the proximal pair of blue and yellow amino-terminal domains. There are substantial amino-terminal domain and ligand-binding domain interaction for red and green. In contrast, extended loops connect the two domains for yellow and blue. Switch of dimer partners from the amino-terminal domain to the ligand-binding domain. Two-lobe ligand-binding domain, and the ligands bind to the interface between the two lobes and closes them. Dimer-dimer interface between the red and green subunits of the ligand-binding domains, mediated by a helix, fourfold symmetry at the transmembrane channel domain. The TM1 at the exterior of the channel domain. The M2 helix lies at the pore. The TM3 helix at the inside of the channel, crosses each other near the extracellular surface to take a closed conformation. The TM4 helix at the exterior, interacting with its adjacent subunit. The ligand-binding domain, TM1 segment is extended until it reaches the membrane where it turns and form a short helix parallel to the membrane. The short helix parallel to the membrane may regulate the TM3 conformation upon agonist binding for channel opening. TM3, ligand-binding domain linker adds a turn to the TM3 for red and green, extended for yellow and blue. Distance between P632 of red and green is 27 angstroms, yellow and blue is 50 angstroms. The ligand-binding domain, TM4 segment with a two-turn helix. Distance between C773 of red and green is 69 angstroms, yellow and blue is 33 angstroms. Conformational change of the ligand-binding domain regulates the channel opening.